Next week, we are doing nervous system, and we will start brain. Brain is hard, and unless you are absolutely independently brilliant, don't try to do it by yourself. It's difficult to understand. Here's what I will do for you guys, and I'm going to ask you for something in return. You will not have a quiz next week, except a take-home quiz at the end of the week, if you come to class. If this room is empty, I will A, reserve the right to give you a quiz on Thursday, and B, also not post your lectures. Okay? So do not sacrifice lecture for lab. Okay? So I won't give you guys a quiz next week as long as you show up bodily, even if you are distracted as hell. You'll absorb something, right? And you guys ain't got no excuse because you got your lab practical tomorrow. Who else has their lab practical tomorrow? Aw, you poor bastards. Sorry about that, okay? Taking one for the team. You can tell them how it was. They're going to change all the questions anyway, so you can share information if you want to. So anything else I'm forgetting to tell you? Are there any open labs Saturday? No. No open lab Saturday. Okay, so I think that we got to here, right? Roman numeral 3, which is nervous tissue? Yes. Okay, so here's what we're going to do today. We're going to start to put together what nervous tissue looks like with how it functions, okay? So not just looking at histology, because we all know that you guys are done thinking about histology, right? But we'll start making, you're like, oh yeah. Okay. So let's talk globally about nervous tissue. Stop talking, it's my turn. Okay, let's talk globally about nervous tissue for just a second. So when we are looking at nervous tissue, we know that it's primarily the neurons plus the supporting cells, which are called glial cells, right? And let's talk about neurons first, and then we'll get into glial cells. What is interesting about neurons, and how are they similar and different from other cells? So we got our two neurons right here, Mary Kate and Ashley, with their big support staff, right? So neurons have an incredibly high metabolic rate. They're really, really energetically expensive cells. For those of you who have had phys or are in phys, you know that what they do is they send electrical signals, action potentials, and they're really expensive to maintain. Because of that, they require a constant supply of both oxygen and glucose. And what do you use oxygen and glucose for? Cellular respiration. Thank you, phys students. Okay, so use it for cellular respiration. And so a couple of special things about neurons. First off, they can receive a stimulus, whether it's a chemical stimulus or my eyeballs are receiving a photon stimulus. I don't know what the hell a photon looks like, but photon stimulus, and they're taking that stimulus and they're converting it into an electrical stimulus that goes 250 miles an hour down the fastest axons. So the only other cells that can do that can receive a stimulus and convert it into an electrical stimulus, are muscle cells. So you got muscles and neurons that can do that. And for those of you who care about the phys, the reason that those two tissues can do that and no other tissues can do that is because those two tissues have voltage-gated ion channels on their cell membranes. For the rest of you who are in anatomy, blah, 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 blah. Doesn't make any difference to you until next semester. Okay. And then what's truly special about neurons is that they can take that electrical stimulus or action potential, which is what AP stands for, and they can force another cell to have an action potential or an electrical stimulus because they can release neurotransmitters. They're the only cells that release neurotransmitters that can force an electrical stimulus on another cell. And then a couple of other things about taking care of your neurons. Your neurons are amitotic after fetal development. As a matter of fact, most of your neurons don't even have centrioles, so they couldn't divide if they wanted to. Now, there is an exception to that, because in biology, you know there's always a freaking exception. There are a few neurons that they found in a couple little places in the brain that still can go through mitosis, but pretty much just depend on losing them if you damage them. So they're amitotic after fetal development, so they have to live as long as you live, right? So they can live up to, I think the oldest person's 123 or 126. That does not sound pleasant to me. Like, who the hell would you hang out with? I don't even know. Like, who are you going to reminisce with?
Yes.
There's no, everybody's dead. I remember where that one person, oh, they're dead. I remember that one person dead. So they have to live for as long as you live. And now we're going to get into the functionality. So ask questions if you need to. I'm going to force you to use terminology as often as I can because that's the only way to reinforce nervous system terminology. So first thing is I want you to know that neurons during development had to decide their functionality, and they don't change after that differentiation. So neurons have three possible functions. Neurons can be sensory neurons, in which they're always carrying information toward the central nervous system. Yeah? Sensation. They can be interneurons that are always doing integration, or they can be motor neurons carrying information out. And they're never double duty. A neuron will never do two things after differentiation. So I want you to look at this example here that we've got. We've got a woman right here who has an itch on her skin, and then she responds by scratching it. Does that make sense? Okay, so I want you to look at these three different categories of neurons. Now, I acknowledge that this one does not look like a neuron because I haven't taught you the different structural variations of neurons yet, but trust me, it is. Okay, and I want you to look at these three, and we're going to identify them functionally. We're going to look at what they're doing. So I have three choices. I have an afferent or sensory neuron, I have an efferent or motor neuron, and the last one is called an interneuron because it's between two other neurons, right? And it's going to do integration. So this first one right here is sensory or afferent, exactly. This one is an interneuron, and what is it doing? Integration, exactly. And in this instance, integration is really simple. It's only one neuron, so it's like a 1 or a 0. Do you respond or not respond, right? Of course, your brain is boatloads, right? And it's not as simple. Okay, and then this one going out is a motor neuron or efferent. Yes, efferent neuron, which is how you pronounce it. Okay, so let's talk about this. So I want to add a little bit more detail because you can actually tell me more about those sensory or motor neurons, can you not? So this one is actually responding to an itch on the skin. So it's specifically a blank sensory neuron. Somatic sensory, responding to an itch on the skin, right? This one's still an interneuron. It's not a whole bunch of different categories right now for you. And then this one is responding by stimulating skeletal muscles. So it is a blank motor, somatic motor neuron. Does that make sense to you guys? Okay. So I'm going to reinforce this terminology as often as I possibly can. Okay. One last little question to you. This part structurally is part of the CNS or PNS? CNS. But after you leave that, you are part of the PNS. Exactly. Okay. All right. So now what I want to look at is the structural classification of neurons. So you can look at what a neuron is doing, and you can look at how it's put together. Ready? So there are three different categories structurally of neurons, and you've only seen one of them before. This one right here is what most neurons look like, and it's called a multipolar neuron, and it's the most abundant type of neuron. So let's talk briefly about the other two types and why they're named what they're named. So you're just looking at these structurally, not functionally, and you name them for how many processes are attached to the cell body. 1, 2, don't want to count them. A lot, right? Okay. This one, weird looking, is called a unipolar neuron because it's got one process sticking out from the cell body. It's weird looking, but nobody actually said that the cell body had to be in between the dendrites and the axons. So what this one is the cell body sticking off to the side. The dendrites are still dendrites, but they communicate directly with the axon. Does that make sense? Okay. And then this guy right here is bipolar, in which you've got dendrites that lead in and axon that leads out on the other side, right? And then this one is the one that you guys are used to seeing. The dendrites, there are multiple, yeah, and they all carry information which direction? Toward the cell body, and then one single axon leaves. Does that make sense? Okay, so these are called multipolar. So we're going to go back again, and we're going to do this time, and ask you different questions. I'm going to look at these three neurons, and instead of asking you their functional category, I'm going to ask you their structural category. You ready? So this first one right here is what? Structural category. Unipolar. Okay, let's talk about why that is in just a second. I think somebody asked me about it last time. And then this one is what structural category? multipolar, and this one? Multipolar as well. Multipolar are the most common. The least common are the bipolar. You find them like in the retina of your eye, not a whole hell of a lot of other places. So let's talk about why this one, this sensory one, is a really good one to use. A unipolar one is a really good one to use. So I want to talk about sensation for just a second. So we had somatic sensation. And with somatic sensation, if I used a multipolar neuron for somatic sensation, then where would the cell body be? Where would the cell body be? If, like, the cell body is near the dendrites and the receivers, so it would be out here, right? That doesn't sound very safe for an amitotic cell. So can you see that if you're doing sensation, a much better structural category of neuron to use would be the one that gets the neuron cell body all the way close to the central nervous system so that you can protect it with bone and maybe even with some meninges around it. Does that make sense? So those unipolar neurons are really good ones to use for sensation because you can get the neuron. Now this neuron isn't in the CNS, but it's very close to the CNS and it's more protected. It's right by the spinal cord. Does that make sense to you guys? Whereas this multipolar one right here is fine to use for muscle contraction because the neuron cell body is protected in the central nervous system. Is that okay to you guys? Okay, so that's a unipolar one. So question for you. Structurally, I'll tell you that the most common neuron in the brain, structurally, is multipolar. Now you guys tell me what's the most common functional category of neuron in the brain. Almost the entire brain is made of which functional category? It does integration. What are the neurons called? The interneurons, because it's all neuron communicating with other neurons, right? Really no sensation to speak of, no motor control to speak of in the brain, right? So the brain is just a big old blob of interneurons. And notice that this figure that we started looking at last time, this one right here, this was a chunk of brain tissue. What structural category are those? Multipolar. And functionally, they're communicating with one another, so they are interneurons, yeah? Does that make sense? So your brain is mostly that. Okay, so, and we will reinforce that as we actually go through. So let's go and look at the structure of a multipolar neuron. Since they are the most common ones, let's learn those in a little bit of detail. And it's not hard because you already did a little bit of it in histology. Okay, so here we go. This one is structurally a what? Multipolar. Can you tell by looking at this particular image what it's doing functionally? You can't, can you? Because it's not attached to anything, right? So if it were going to a muscle, it would be motor or efferent. If it were coming from a sensory receptor, it would be sensory or afferent. Or if it had two other friend neurons, it would be an interneuron. But this one in isolation, you can't tell, right? You don't have enough information to tell what it's actually doing functionally. Go ahead.
What's the axon collateral coming off the side?
I'll tell you about that in just a second. I'm going to get to axons in just a second. Okay, you guys ready? Okay, so you understand why you can't tell? You actually have to hook it up to something in order to figure out what it's doing. Okay, so let's look at the structure. So first off, the cell body, of course, which is where most of the organelles are, right? And the organelles, the only unique thing that you guys need to know about the organelles is that usually the nucleus and the nucleolus, I don't know if you can quite see it from there, the nucleus is lighter and the nucleolus is darker. Let me just show you this histology image. So what happens with neurons is that you get this owl, they call it an owl eye nucleus. Do you see that? So that like the little pupil is really dark. So it's just like characteristic that you can see sometimes, not in all cells, but in most of them. And then the other thing which is interesting both structurally and functionally is that notice that the cytoplasm looks really grainy, like granular. The reason is because these are called chromatophilic substances, but what it really is boatloads of rough ER, because most neurotransmitters are amino acid based. Yeah? And so, does that make sense? So they're actually going to have to do a lot of amino acid packaging, synthesis, make little bitty peptides and that kind of stuff. Okay, so that's the chromatophilic substance, which is just the rough ER. Okay, so let me back up just a little bit and say that whenever you have a whole bunch of neuron cell bodies. In the CNS, it tends to make the tissue gray, right? So you got gray matter. And when you have gray matter around the outside, it's just called the cortex. But you also have blobs of gray matter on the inside. Whenever you have a blob of neuron cell bodies collected with its buddy inside the CNS, it's called a nucleus. Okay? So all of these nuclei have individual names, and you'll learn some of them later. I don't make you learn them all because there's too damn many of them. Okay, so this is a nucleus, okay? But when you have a blob of neuron cell bodies outside the central nervous system, it's called a ganglion. So this guy right here, he's not hanging out by himself. He's got a whole bunch of his buddies in there. It makes a little blob of neuron cell bodies, and that's called a ganglion, okay? And I'll reinforce that as we go through, okay? All right, so now the neuron processes. Okay, so these are dendrites, and you know that dendrites receive information, carry it toward the cell body. Okay, so what do I mean by receive information? Well, let's put this together with 1610 a little bit. They receive information because they have cell membrane receptors on the dendrites, right? And so what can happen is it can respond to, for instance, chemicals that will release by another cell, respond to them, and then send that impulse toward the cell body and out the axon, right? Or it can respond to light, right? It's got light receptors on the dendrites, right? So these are receiving information of various sorts, and then what they're doing is sending it toward the cell body, and then what the cell body can do is with the axon, it can actually convert it into an electrical stimulus and send it out 250 miles an hour. That's what it can do. So the dendrites are receiving and the axon is sending, okay? With a multipolar neuron, you can have multiple dendrites. With a single, like I told you guys last time, with a histology image, I don't think you can really tell an axon from a dendrite unless you can follow it for a really long time. So it's kind of arbitrary to label those. But if you have a picture, you can, right? A drawing, you can. Okay, so let's talk about the axon, and we'll talk about the axon collateral. So with a multipolar neuron, you only ever have one axon attachment to the cell body, but there is literally no limit to how many times it can branch after that, okay? So the axon hillock is the point of attachment to the cell body. It's really physiologically important and interesting. It's the thing that it can actually translate whatever input you got into an electrical impulse. Okay, now the axon collateral is a branch from the axon after it left the cell body, and there is no limit to the number of axon collaterals you can form. As a matter of fact, one of the things that we've learned within the last 15 years or so is that what is going on in your brain when you are learning new information is that you are actually changing the branching pattern of your axons. You're not getting any new neurons, but you're changing the branching pattern. So for instance, and this is a metaphor because it's not just one branch that occurs like this. You have known for ages that if you work out in a certain way, you can build muscle mass. Yes? You've known that for ages. And then you learned in this class, perhaps for the first time, that how you actually build more muscle mass is that each cell gets more myofibrils in it, right? So there's two packets of information that were unconnected, right? And so what actually happens when you learn is you actually make an axon collateral that will connect that packet of information to the other packet. When you learn, you are actually making more axon branches in your brain. You are forcing what we call synaptic plasticity. You are changing the structure of your brain. So learning, an awful lot of learning, is making new branching patterns in your brain. And most of those branching patterns will be myelinated, so you actually end up making more white matter in your brain. Even though you can't get any more cells, you can't really get any more gray matter, but you can certainly change the amount of white matter in your brain. Does that make sense to you? Now, it's not a one connection thing. That was an oversimplification. But it's basically packets of information connected to other packets of information and then making connections between the two of them. Does that make sense? Okay. All right, so that's an axon collateral. There's no limit to the number of axon collaterals that you can make. All right, so, and then if you follow the axon, not all axons branch this way, but they will all branch at the end, okay? At the end, when you get to the end of an axon, you will come to what we call axon telodendria. So I am the axon cell body here, and here's my axon, or neuron cell body. This is my axon, right? The fingers at the end are axon telodendria. Telo means end, dendro means fingers, so it's literally the fingers at the end of the axon, right? Does that make sense? So an axon, here's my axon, here look, yes, connected to my cell body. Here's my axon telodendria. And then the fingertips, notice, look like little ET fingers, yeah? Why are they bulbous like that? What do they store in there? Neurotransmitters, that's where neurotransmitters are stored. Unfortunately, nobody can decide what the hell those things are called. You can call them synaptic knobs, synaptic terminals, axon terminals, terminal bouton. Doesn't matter, I don't give a crap what you call them because it varies too much. I do care that you know that they're at the end of the axon telodendria. They're going to store and release neurotransmitters. Unfortunately, your phys book is going to call them something totally different than your anatomy book. I don't know why the hell they didn't get it together. But there is no consistency in any textbook about what they call those. Okay, I tend to call them synaptic knobs or axon terminals. All right, so let's talk about the myelin sheath that you can actually see there. So this myelin sheath looks like sausage links along the axon. Yeah, do you see it? And here's what the myelin sheath does, and I'll add some clinical information for you guys to make it make sense. The myelin sheath is for insulating and making that electrical stimulus go faster down the axon. So I want you to think about this metaphor. When you were doing a science experiment when you were a kid, or with your kids if you have kids, and you're trying to make the potato battery light up the light bulb, trying to do it, you got a little copper wire between them, right? If the potato and the light bulb are really close to one another, do you have to insulate the copper wire? No, you don't, right? So as long as it's relatively short, you can depend on the electrical stimulus getting from point A to point B. But as it gets longer, it's more likely to come into contact with other things. If you're making a Rube Goldberg machine out of the damn thing, right? Because you got a smart kid, or you're doing their science experiment for them, don't do that. So when you're doing that, then what are you going to have to do in order to get the stimulus to go all the way from the potato to the light bulb? You're going to have to insulate it. Okay, same thing with you. If I have two neurons that are right next to one another, insulating the axon is unnecessary. If, however, I have a neuron that's in the lumbar region of my spinal cord that's going all the way, the axon's going all the way down my leg to the muscles in my little toe, if I do not insulate it, then the stimulus will not get down there. That is what happens with multiple sclerosis. So MS, or multiple sclerosis, is an autoimmune disease that causes demyelination of the axons. And let's think about where it affects. Do you guys know anything about MS, where the symptoms tend to show up first? Hands and feet. Why? There's a lot of myelination that's necessary. And if I lost just a little bit of it, do I actually get it all the way down to my hands and my feet? No. Does that make sense? So the longer the axon is, the more necessary it is to myelinate it, and the more symptoms you'll get if it gets demyelinated. Does that make sense? Okay, so let's talk about how myelin works. Okay, so if you look at this figure, you can see that in this particular instance, myelin is actually like little cells that are wrapping around like sausage links. So let me show you how that works. This is how myelin works in the PNS. It's slightly different in the CNS. Okay, so what I have is an axon, and then I have one of the glial cells that's going to devote its entire life to wrapping around and around and around that axon like a jelly roll. Oops, lost it. Wrapping around and around that axon like a jelly roll. Do you see that? And so really what myelin is layer after layer after layer of cell membrane. Yeah? Because you've squished out almost all of the other stuff to the side. And although an individual cell membrane is clear, like saran wrap, is a whole roll of saran wrap clear? No, it's opaque, right? So what you end up getting is wherever you have myelin, the tissue looks white. Yes? So you will get white matter wherever you have myelination. So this is a myelinated axon in the peripheral nervous system. I'll show you what it looks like in the CNS. Let me show you a couple more pictures just to reinforce it. This is a different type of microscopy than you're used to looking at, but this is a myelinated axon. Do you see? Layer after layer after layer of myelinated axons. Pretty cool, huh? And then this figure that you guys looked at last time, you didn't really know how myelin was formed yet, but we can look at it again. So remember that this was the endoneurium, right? And now do you see what this is? That's layer after layer of myelin wrapping around, right? Okay, so this is basically a cross section of one of these sausages. Yeah, cross section of one of those sausages. Right. Okay. So now, when you myelinate in the PNS, you use these cells that we just talked about. These cells are called neurolemocytes or Schwann cells. Neuro meaning nerve, lemo meaning layer, as in laminate, right? Layers. Site, cell. Okay, so that's the way you do it in the PNS. In the CNS, you accomplish the same thing with a different kind of cell. So here's PNS. Yeah, that's how you myelinate an axon in the PNS. And then here, how you myelinate an axon in the CNS is with a different cell. The cell body sits off to the side, and it extends fingers of cell membrane to wrap around and form little sausage rolls. This one is called an oligodendrocyte. Oligo meaning many, dendro meaning finger, cell. Yeah? But you accomplish the same thing, right? Okay. Now, why the hell do you care which cell? It actually is clinically important. Because if I damage this axon in the peripheral nervous system, these cells will try really hard to make a little tube that guides the regeneration. It's called a regeneration tube. And if you damage white matter in the central nervous system, these cells just go, I'm out. They don't help at all, right? So I know that you know this, but reason number 452, that if you had to damage nervous tissue, would you rather damage PNS or CNS? PNS, exactly. None if you could choose, but definitely PNS before CNS. Okay, so let's just think about that for just a second. Now I want to ask you just a little bit about this. So let's go back to this figure that we looked at last time, this really cool figure. So what I want to know is, why is this sciatic nerve right here, why is it white? What kinds of cells are responsible for that? It's the myelin sheath, but what kinds of cells? Because you are in the CNS or PNS? Neurolemocytes or Schwann cells, okay? But now let's look at this figure that we've looked at before. Okay, I want to know why this white matter in here is white. Oligodendrocytes, yeah. So it looks the same. Clinically, it has different significance. And by the way, the two different cells have different predispositions for cancer, too. So clinically, it ends up being important. There's like oligodendromas, and there's schwannomas, or neurolemomas. Okay, so white matter and gray matter, again. Remember, gray matter is neuron cell bodies. White matter is basically wiring, right? Isn't that what I said last time? It's wiring. It's myelinated axons. Okay. So now, with that in mind, you should be able to very easily look at this cell. And you don't have to do it right now. I just want to see if you can do it. So the good part about learning the A of nervous system is that it gives you a preview of the P. So I want you to look at the cell and say a couple of different questions. Can you tell me the structural category of this neuron? What is it? Multipolar. Can you tell me the functional category of that neuron? No, because it's not attached to anything. Can you look at this neuron and tell me the direction of information flow? Yeah, it's going that direction. Good. Okay. Label the axons and the dendrites and the neuron and the cell body and the axon telodendria. Right? You can do all of that. Now, can you tell me whether this axon is going through the PNS or the CNS? Who can tell me? Go ahead. And how do you know, Terry?
There's a couple ways to tell, but one of them is by looking at the Schwann cells.
Yeah, the Schwann cells, right? If it was CNS, it would be myelinated a different way. Does that make sense? So there's a tremendous amount of information you can have, and here's the axon hillock. One last thing that I didn't point out to you before, let me point it out. Regardless of whether you're in the CNS or the PNS, there are always little unmyelinated portions between the sausage links. Those are called nodes. Nobody's decided which kind of node. They're either called neurofibril nodes or nodes of Ranvier. It doesn't matter, because if you see the word node, that's what it's talking about, right? There's only one node in this story that's relevant, okay? And these physiologically are really important. It's hard to explain what they do in anatomy, but in phys you'll learn what they do. Okay, so now let's talk about a synapse, okay? So what happens when you get to the end of the axon? When you get to the end of the axon, of course, you can figure out that you're going to release neurotransmitters, right? But what you're going to do is you're going to release them right next to the cell that you want to talk to. And the cell that you want to talk to can be another neuron. If it's another neuron, then you communicate neuron to neuron, and that's called a true synapse. But neurons can also communicate with other kinds of tissue. What other kinds of tissues can neurons communicate with? Muscle and glands, right? So if it's a neuron to muscle synapse, it's called a neuromuscular junction. If it's a neuron to gland synapse, it's called a neuroglandular junction. Yeah? But we're just going to talk about neuron-neuron synapses, which are often called true synapses. So we're going to talk about those today. Those things are complicated as hell. For people who have taken physiology, you know that there's a lot more to this story. For right now, we are just going to focus on what's going on at the synapse, just anatomically. So really what a synapse is the junction between a neuron and a neuron, or a neuron and an effector. And basically it looks like the synaptic knobs or axon terminals or whatever are touching the next cell, but they're not. They're actually microscopically separated. So don't worry about learning this figure in detail because it's a phys figure, but I just want to give you a preview of things to come. This is the little ET finger at the end, right? And this is the next cell. So what's actually going to happen is you actually have the capacity to release neurotransmitters into the interstitial fluid so that you can communicate between the two cells. And this gets really interesting in physiology because this is how a lot of your neuroactive chemicals work, right? Like, for instance, an SSRI will just make sure that if this was serotonin, it gets to stay there longer. If it's LSD, it just pretends like it's serotonin, right? And it makes you hallucinate. So there's a lot of really interesting phys that happens at the synapse, but for right now I just want you to know that you almost touch the next cell, right, with the synapse, and then you release chemicals to communicate between the cells. So with that in mind, you should be able to look at a simplified figure. Now this is two neurons in series, and you should be able to tell a few things instantaneously. Which direction is information flowing through these two neurons? From that direction to that direction, right? And so with that in mind, nobody would have to tell you which neuron was the presynaptic neuron and which one was the postsynaptic neuron, right? That makes sense? Presynaptic, postsynaptic neuron. And you should be able to label an axon hillock on one of them and an axon on both of them, right? Cell body and dendrites on one of them. Yes? You should be able to label axon telodendria on one of them, the presynaptic one, right? And the axon terminals, and you should be able to figure out where the neurotransmitters are released. So again, the really good thing about putting some time into nervous system anatomy is because nervous system physiology is where it actually gets hard. And what you want to do is to have like half of the answers filled in when you just look at the image, right? In anatomy or in phys, because you're like, oh, I got this. I know exactly which direction things are going. I have a prediction about how things are going to occur. Does that make sense to you guys? Okay, so I'm going to let you guys go a little early, but I think I'm just going to let you go early because they're so far behind you. So what we're going to do next time is we're going to finish up this set of notes, and then we will start the brain on Tuesday, okay? No in-class quizzes next week unless nobody's here on Tuesday. Have to manipulate people into coming to class, damn it.